upset and angry because they use all of these little traits, right, to differentiate between, to identify which species is which. But in general, like, for the most part, at least in the flowering plants, they all kind of look the same. They're flat and they're green, right? They're just a green, flat sheet. And unless you start to look at other species, like the conifers, that are kind of round and pointy, right? You can basically break leaves down into the either thick, like maybe a cactus, right? So that's just kind of a big leaf. And it's sort of a barrel shape. But then if you start to get away and look in kind of temperate regions and outside of the extreme climates, everything is sort of following this same pattern. It's just a flat green sheet. And that raises an interesting question. And even if you start to look at all the different leaves, and you look at compound leaves, and you look at all these leaves with different margins, they're all sort of still following that same flat green sheet template, which I think is really interesting. And then so that leads us to a very broad question. We'll kind of narrow down our questioning as we go through the talk. But that leads to a very broad question: is why does a leaf actually look like a leaf? Right? Given the amount of species diversity, 400,000 different species of plants, and sure, you can have a really large leaf, right, up a meter squared, and those big elephant ear leaves that grow down in the tropics, or you can have one of those teeny tiny little scale leaves that's on a juniper, for example, right? Several orders of magnitude in size, but in essence, they are all just a flat green sheet, right? Thin, flat sheet of green, right? Why, right? Why is that? So let's just take a look at one leaf, and this is interesting that Graham, I didn't know that Graham was going to introduce me today until he got here, and Graham has been working on red maple for a really long time, and red maple is just the leaf that I decided to choose to just take a look at. We're going to just pick one of those leaves, and we're going to look at it carefully and think about why a leaf might look the way it does, right? So it's all the flat green sheet, and there's some differences in kind of what the leaf margins look like. But in general, right, there's the shape and the form of a leaf is relatively constrained. Given if you look at animal systems, animals look really, really different, right? But when you look at the leaves in a forest, they're all kind of the same and all working from the same format. I understand that there's a lot of important variability here, but for the sake of argument and following along with me, we'll just think about what it takes to be a leaf and how you go about building a leaf moving into the future. And I'll talk a little bit why thinking about things in this way are probably going to be important long-term for us as humans living on a planet with perhaps 11 billion of us sharing the exact same resources with a very limited set of species that we like to eat. All right, so a leaf needs to be good at being productive, right? So a plant is going to spend, there's going to be some cost that goes into making this leaf. It needs to be good at light acquisition. It needs to be good at energy conversion, right? So it's pulling photons out of the sky and then turning that into something that the plant can use, right? So a plant needs to be good at that. And a plant also needs to be good at transporting things, so moving things around. So it needs to be able to transport water into the leaf. If you start to look at leaves carefully and hold them up to the light, for example, as everybody walks out of the room and sort of walks and goes on their way, pick up a leaf and hold it up to the light. And you'll see this really intricate venation network, right? So this is the transport system of the plant that's delivering all of that water. There's 400 water molecules for every CO2 molecule that's coming in, and that CO2 has to come out, right? In the form of sugar, it's got to get out of the leaf, right? So this is just a transport problem. And so even if you're not a plant biologist or a physiologist or a biologist or an ecologist in any way, if you're somebody who's into urban planning, for example, we can learn a lot, probably learn a lot from leaves, right? So this is the vascular system of a red maple leaf zoomed in, and this is a city map of New Haven. Right? So you have the same thing. You've got a big major vein, and then you've got some secondary veins that go out and deliver water exactly where it needs to go. This is I-95 kind of running through. Q Bridge is over there somewhere. And I'm not saying that New Haven has the best highway system, right? But plants have a several hundred million years advantage in figuring out how to distribute resources in a very efficient way. But if you go through New Haven, you can get off the highway, and then you get off onto these side streets, and then smaller surface streets, right? So there's a lot of parallels actually, and probably a lot of things that we can learn from plants, and in terms of distributing resources and moving things around. Okay? So a plant needs to be good at those a few different things: getting water in, getting carbon out. But at the same time, it's got to be good at doing a lot of other things. And I think one way to look at a leaf, and the reason that a leaf looks the way that it does, is you can think of it in terms of a plant and its sort of journey through evolutionary time and the resolution of the sort of these tensions that arise between kind of a multi-dimensional demands of leaf form and function, right? So it's got to be good at leaf absorption, so it might be getting tugged in a certain way through natural selection, and it needs to be flexible, and it needs to be durable, as we all saw yesterday with the wind that was blowing around and the rain that's coming down, right? Those leaves are flopping around, and there's a lot of kind of mechanical trauma that's happening, and then as soon as the wind dies down and the sun comes out, and so there's all sorts of kind of mechanical constraints, and they also have to be good at not being eaten by other organisms, right? So leaves have to do a lot, right? And they have to do a lot of different things. And what we're trying to do is figure out which one of these is most important, or what are the kind of the constraints of putting together a leaf, and whether or not a lot of these traits that we actually look at in leaves are meaningful. And Marlies, I think she's here. So Marlies and I get in this argument all the time about whether a trait is functional or not, right? So her students are in dendrology, and you're trying to key out different species and say, okay, this particular species has a lot, toothy leaf margins. Well, does that actually mean anything for the physiology of the tree or not? And so I think there's a lot that we can learn from each other in kind of thinking about leaves in very different ways. And one of the problems, I think, with our field, plant physiology sort of broadly under that big umbrella, is that the people that study water transport, or the people that study CO2 absorption, or light processing in particular, don't often sit in the same room. They're not communicating to each other. And so what we've tried to do is kind of curate a way of bringing together lots of different people, lots of different labs that have very specific questions about the way that plants work, and thinking at bringing them together. It's sort of like another version of FES, where we have people from many different fields coming together, thinking about the environment from very different perspectives. Okay, so how to build a leaf, right? So it needs to be good at photosynthesis, capturing light, moving water, acquiring CO2. It needs to be good at getting that sugar out. This is something that people often forget. We often focus on just, the leaf veins are really, really interesting, and its patterns, and they're really, and the leaf and the vein density and things like that are really important for transporting water, but they're almost equally important for getting the sugar out. If the sugar doesn't get out of the leaf, then all of the water import doesn't matter. They also need to be self-supporting structurally, and we'll talk about that towards the end, which is actually a really cool area thing that we're working on recently, and really neat interdisciplinary project. And then they also need to be cost-effective. So in terms of an economist, there's kind of been a running narrative for the last decade or so on this leaf economic spectrum. So thinking about leaves in terms of how much do they cost, how much do they cost to operate, and then how much do they bring in? What's the net gain? What's the net gain for a tree in terms of the carbon, water, and nutrient investment, right? So thinking about all these things, and this is all the different constraints that a plant is under in putting a leaf together. Okay, so what constrains leaf form and function? And we can consider two different possibilities, right? So just two for the start, and thinking about these two properties, and then trying to think about looking at a couple of different case scenarios. And trying to figure out why leaves look the way they do. So this first consideration here is that 400-to-1 ratio, right? So plants are going to need a lot of water in order to pull in CO2. So we're going to think that leaves are probably going to be built around, or leaves that are going to be more and more efficient over time are going to be built around putting together a vascular system that is very, very efficient, right? So the more water that a leaf can pull into the more water a leaf can pull into it and then out into the atmosphere, the more CO2 it can gain. And the other one that's really interesting, and this is just a fundamental biophysical principle, that CO2 diffusion, so the diffusion of carbon dioxide, is 10,000 times slower in water compared to air, right? So if you have CO2 bouncing around in the atmosphere here, it's the diffusion of CO2 into my liquid water here is going to be the diffusion of CO2 through that liquid water is going to be 10,000 times slower. So there's some things that this is a really big fundamental biophysical constraint that is probably pushing plants to look the way that they do. All right. So one of the interesting things that's happened, and this comes from the kind of the paleo literature, looking back many millions of years ago when plants emerged from the oceans, right? So they originally started in the sea, and then they came onto land, and they ran into all sorts of different problems. So access to CO2 was now much easier, right? Because if you're in the water, the diffusion of CO2 is really, really slow, and it takes a lot to pull any CO2 out of the water. And this is sort of what we call the Lilypad principle. So if you look at a lily pad, it's sitting on the water, and all the stomata are on the upper surface. There's no stomata, the pores on the surface of the leaf that draw CO2 in. The pores the stomatal pores are all on the top surface of a lily pad, because the bottom side is covered in water, and the diffusion rate is so much lower, right? So the lily pads have figured out that they can do much more photosynthesis and have a really, really big leaf by having all their stomata on the top, right? And so one of the trends is that 400 million years ago when this happened, there was really high CO2 concentration in the atmosphere, the temperature was really high, and plants had few stomata, and overheating was a really, really big problem. And so over time, about 360 million years ago, CO2 is starting to decline. The CO2 concentration in the atmosphere is starting to decline, and the access then to CO2 is a problem. This should be a subscript and not a superscript, and I'll make a note of that to change that. But basically what happened is these very early plant forms, these photosynthetic organisms that were on the planet, started to change. And so this is kind of the equivalent of a duck, right, getting the webbed feet, or Michael Phelps with the extra little skin in between his fingers, right, that makes him swim really, really fast. This basically turns a set of cylinders, kind of bifurcating cylinders. This is what these plants look like millions and millions of years ago, and then transition to something that looks more like the leaf today. And this probably happened around 360 million years ago, and basically every leaf that's on the planet now looks like this, following the exact same template. So plants very early on figured out that this kind of solution, the flat green form, right, the flat green sheet or the thin green sheet, was the way to do it. This is a major advantage, and this worked really well, and any deviation from doing it this way led to disastrous consequences and extinction, probably. Okay, so when we think about water actually moving through the plant, and finally into the leaf, so up through the roots, through the trunk, out to the branch, through the petiole in the liquid form. And so the water is going in as a liquid and then eventually making its way into the teeny tiny veins. This is one of the, a cleared leaf is what we call it. So we've bleached away all the chlorophyll and then stained it. And you can see all the veins that are in there. The water is going to go through one of these, the mid vein of the leaf that we're all very familiar with. And then if you take a leaf and you look at it underneath, just hold it up to the light and you'll be able to see it. You don't even need a microscope. You can see all these teeny tiny little pathways and these little veins. And what they're doing is the veins are transporting the water through a relatively low resistance pathway. This is just pipes, plumbing, that's very, very efficient. And then the water emerges from one of these veins. This is kind of looking at it from a 90 degrees view from above. This is basically from above, and if you turn that plane 90 degrees, you're looking at the xylem, right? So we remember xylem and phloem from our introductory biology classes many, many years ago. And so there's the xylem, and then the xylem is transporting water in the liquid form, and then it has to diffuse and basically seep out from the vasculature through all of the mesophyll cells. And these are all the cells here, and all these little jelly beans that are kind of scattered around here would be green under normal conditions. And those are the chloroplasts. Those are what are doing the photosynthesis. And this is where the water has to go through cell to cell, and it's very slow and inefficient. And then eventually it gets to this surface, right? This surface here, and then it makes the great leap from the liquid phase to the vapor phase, and then eventually diffuses out of the leaf through the stomata. Right? Those are the little teeny tiny pores that can open and close that allow water vapor out, all those 400 water molecules out for every one CO2 molecule that comes in. And then CO2 basically goes through the opposite pathway. CO2 goes in, is absorbed by one of these cells, converted into sugar, which then has to go back through the same pathway and then get into the vasculature, but not in the xylem, but in the phloem instead, and then transported out of the leaf, right? So this part of the pathway ends up being really important. But this part, so how many stomata there are, the density of the stomata on a surface, so basically the number per square millimeter, is going to be very important for allowing water out and CO2 in. The vein density is going to be very important as well in terms of getting the water as close as possible and sort of minimizing this high resistance pathway as possible. And that's actually what happened, sort of a trajectory towards that. Right? So if we look at time on the x-axis here, so this is deep time. We don't normally think about time on this unless you're into the paleo world. But we're talking, what we're interested in is sort of the last 200 million years, right? And so if we go back 200 million years ago, so we're fast forwarding from those really weird kind of odd leaves. If we go back 200 million years ago, and we go out and do a census, a survey of all the plants that are on the planet, and this is biodiversity on the y-axis, or the percent of the species, we're mostly looking at ferns, so the pteridophytes and cycads. Ferns and cycads would be a really weird looking place compared to what we see today. Right? So the ferns and the cycads dominated in terms of species diversity and probably abundance too. And there were a few seed plants, and then there were a lot of conifers, right, there. And then over time, what happened is this explosion of the angiosperm diversity, right? So the angiosperms in green here, and going through time, and in particularly right around the Cretaceous. And then there's a second explosion right around the boundary between the Cretaceous and the Tertiary, right? This is what we often refer to as the KT boundary, where there was an additional explosion in the angiosperm diversity. And so one of the questions is, well, how? How did they do it? So you have these other plants, right, the ferns and cycads and conifers that were dominating the entire planet, And then in a very brief period of time, in terms of evolutionary time at least, the angiosperms completely take over the plant. So it's a really compelling story, and we don't have all the pieces put together. What we know is happening at that time, depending on the model that you look at and the proxy data that you use, what we know is that during the Cretaceous, during this green sort of highlighted area, is that CO2 concentrations were going down. I mentioned that. And in response to that, one of the really interesting things is if you go back and you look at the fossil record and you can kind of reconstruct when different events happened in terms of the evolution of the leaf and what the leaf looks like, is stomata went from being really big to really small. And they also went from being fairly few stomata to lots of stomata. Right? So remember, the stomata are the pores that are letting the CO2 in and the water vapor out. And it turns out that small stomata open and close much faster. Right? So if water is limiting, the plant wants to be really careful with how much, very conservative with how much water it uses. But when they're open, they want to be as efficient as possible in letting as much CO2 in and as much water out. And so as you go through evolutionary time and you look at all the different species, the diversity in stomatal, the stomatal conductance goes up. And the stomatal density goes really, really high, right? So plants are taking advantage or figuring out that they are, one of the constraints is that the CO2 concentration is dropping. And in response, they basically poke more holes in the underside of the leaf. And the other thing that they did at the same time was to increase vein density. So if you go way back, the density of the veins, so if you were to take a square millimeter or square centimeter of vein and sort of trace out the total length of vein that's there, basically what happens is that there's low vein density back in these early plants 150, 175 million years ago. And then over time, vein density has increased, is sort of the general trend, right? So more and more veins mean the water gets closer and closer to the sites of evaporation, and it means that this kind of high resistance pathway is minimized, and that will allow more water to get out of the leaf, and then the plants to do more carbon fixation. And if you can get more carbon into your plant, you can make more leaves, you can make more seeds, and you basically come up with sort of the dominant solution, and this is what the angiosperms did. But what we realized not too long ago is that there's a big missing piece. So we knew that in this leaf hydraulic pathway that I just showed you, the water goes in through the vein, it passes through the mesophyll. These are the cells that are doing photosynthesis and where all the evaporation of water is going on and the absorption of CO2 is going, goes through the mesophyll CO2 and water exchange while satisfying all those other demands that I talked about, right? So if CO2 is dropping in the atmosphere and more CO2 into the plant means a better plant and a more competitive plant, then how can a plant modify itself, modify its structure, sort of its architecture, in order to optimize the uptake of CO2 while maintaining enough water and satisfying those demands, all right? So if we think about the inside of the leaf, one of the classic paradigms of fundamental paradigms of biology is this surface area to volume ratio, okay? So if we think about this, we do this every day. Everybody take a deep breath and exhale. All right, so we're all good. We're all feeling good, right? And so that's just air went into our lungs, right? So the human lungs, this is a marvel of the surface area to volume ratio, right? So if you take the human lungs, 300 million alveoli, or these little parts, and if you take that and you spread it all out, right? So if you took all the surface area and calculated the surface area in the human lungs, the surface area is somewhere in the order of 50 to 70 square meters, right? For one set of lungs, right? And that just highlights the importance of the surface area. So if we have a large surface area, that means we can pull more oxygen into, I don't know what happened there. That means, oh, thank you, it looks better. All right, so that, and it's going to look better. We're not going to look at lungs all day. That means that you can pull more oxygen out of the atmosphere and dump it into your blood, and that allows humans to have a really high brain function and capacity for, I like it off, keep them off. Did somebody bump it back there? I think they did, all right. Oh, that happens, thank you. All right, no, all the way off. All right, so human lung, right? So one set of lungs, 50 to 70 square meters. That's like half of a tennis court, right? In surface area. Ooh, now we're good. All right. Okay, so we thought, all right, well maybe it's surface area, right? So what we did is we went out and we looked through the literature. Laura, was that you again? No, all right. So what we did is we went out through the literature and just kind of did a review and said, okay, what are the maximum photosynthetic rates? So this is in micromoles per meter squared per second. So how much CO2, what's the maximum amount of CO2 that a plant can pull out of the atmosphere for a given surface area? And we're calculating surface area by looking at, how much kind of cross-sectional area we're looking at, and then how much, and then basically tracing the lines around these borders and figuring out how much surface area there is. And there's nothing, right? So this is just, it just looks like somebody just, it's a shotgun, right, of just a huge spread in the data, and very unsatisfying and very sad. And so we thought, oh, maybe this is it, maybe this is it, maybe this is the thing that people have been looking for in order to plug in that missing piece, and like maybe plants are also doing this sort of a coordinated evolution of vein density and stomatal density and mesophyll surface area, with maximizing the surface area to volume ratio. But it didn't pan out. And then so we thought, well, we think we're smart, and we think we have good questions, and so maybe what if we've just been measuring it all wrong, right? So maybe we've just been thinking about this in the completely wrong way, because we weren't able to measure these traits in the way that are actually meaningful for a plant. And so traditionally, the way this is done is you take your cross-section of your leaf, right? So this is the vein in the middle, this is the palisade mesophyll, and this is the spongy mesophyll. This is basically acting like a sponge, which is why it's called the spongy mesophyll. It looks like a sponge and acts as like a sponge, pulling CO2 out of the atmosphere and letting water vapor out. And so what you would do is traditionally you would go in and you'd make all these different detailed measurements of the outlines of all these different cells and add that all up, and that's your surface area. But what we know is that plants don't necessarily look like this. And the reason that we probably got this wrong to begin with, and the way that we're thinking about this in the wrong way, is that this is sort of a two-dimensional cross-section that you would get from a light microscopy slide. So you take a leaf, make a really, really thin section, put it on a microscope slide, and this is what you would see, right? You've got the epidermis, you've got the top of the leaf, the bottom of the leaf, you've got the guard cells that form the stomata, the stomatal pore, and then you've got all these cells, the photosynthetic material in the veins, and then this is like the most detailed one that I could find, this sort of three-dimensional view of what a leaf actually looks like. But it turns out that the three-dimensional view, our cartoon sort of version of the leaf as it's presented in introductory biology classes, is all inferred from a two-dimensional perspective, okay? And we think that that's probably led us down a bunch of wrong and dead ends in interpreting what the leaf actually looks like on the inside. All right, so one of the things that we've done, we've been working on sort of using this new technology to basically do a 3D imaging of plants. And we've done this quite a bit with a number of different plant species, usually focused on roots and stems and trunks over the last probably 10 years or so, and trying to understand sort of the functional status of the vascular network as it's exposed to drought. And then what we started to realize is that as this technology has sort of matured, and as we've refined our methods for doing all the image processing, we can now take advantage and start to look at leaves, and then actually measure the actual surface area of the inside of the leaf for a given volume of leaf tissue instead of a cross-sectional area. And so what we use is it's called X-ray computed tomography or microcomputed tomography, CT scanning, and it's basically just a three-dimensional X-ray, and this is based on the same medical principles, same principles as a medical CT system. So if you had something that happened to your body and you needed to get the doctor wanted to get a three-dimensional scan, they might pair it up with some sort of dye tracer that would allow you to figure out what was wrong with a certain part of your body. And basically what a CT scan is hundreds of standard X-ray images, but just taken at different angles, and a whole bunch of math that allow you to get kind of a three-dimensional image of what you're trying to study. And so we're using the same type of instrument, but it works at a much smaller scale. So the medical CT units are kind of like a big donut, and sort of look like an MRI as well, but they're a little bit smaller, and you lay on this platform and you slide in. So we're basically doing the same thing with plants, except we're doing ours at the Lawrence Berkeley National Laboratory. And this is probably one of maybe 2 or 3 places in the world that can actually do the type of imaging that we want. And so it's a synchrotron particle accelerator facility, and one of these little arms off of here, so there's high energy photons and racing around this ring, and they do all sorts of interesting particle accelerator physics and things like that there. And there's also one of the what they call a beamline here, where they shuttle off some of the X-rays that are produced by this instrument. We can use that to do this X-ray imaging. And so this is what it looks like from the inside. It's about the size of almost the size of kind of a soccer stadium. It's huge. And then in there's this metal hutch. And then inside this lead-lined hutch, because there's a lot of X-rays and we need to be careful, there's a rotating stage. The X-rays come out of here and pass through our sample, which we have a highly sophisticated sample mounting system, which ends up being a plastic pipette tip that costs like a penny a piece, but it turns out to be the perfect sample holder. And this is something that Adam Roddy in my lab and Mason Earls and then Guillaume Thiero-Rencourt have been working on over the past couple of years and developing this method as a way to visualize and kind of see the leaf in 3D. Right? So up until now, we've basically been using two-dimensional methods for asking questions about a three-dimensional problem. Right? So we need a three-dimensional approach to doing this. So we'll take our leaf, this is just kind of a brief overview of what we're doing. We take our leaf, we cut out a piece, and then we take it and we wrap it in some X-ray transparent tape that keeps the leaf basically in its original state. Right? And so in this way, we don't have to do any of the dehydration or any of the other sample preparation that you would need to do with traditional histology and microscopy that might lead to some measurement error. We take that, we put it into our sophisticated sample holder, our pipette tip, we mount it in the instrument, and then we get basically thousands and thousands of cross sections for a given final volume. And the data that ends up coming out ends up for like each leaf, it's probably 5 to 10 gigabytes worth of data. And so data processing and management ends up becoming a major problem for us. And this is something that Guillaume and Mason in particular have been focusing on, figuring out ways to get the computer to do a lot of this work. And so what it's required of us is to basically teach the computer plant anatomy, leaf anatomy, so that it can automatically go through these thousands and thousands and thousands of images and pull out what we think are the important parts and doing it in an appropriate way. And so we get cross sections through the leaf, and it goes through a bunch of different image processing steps. And then we basically have this kind of exploded parts diagram of what a leaf is, right? So essentially for the first time. And what we've got is this is all the leaf material here. This is all the airspace that I was talking about. So it's sort of the inverse of all the leaf tissue, right? And does that look like that cartoony sort of three-dimensional like cartoon of a leaf? No, it doesn't. It looks completely different. We've been wrong, right, for years, all right? Which is surprising. And I showed this to Erica Edwards the other day, and she's in the back, and she just, mind exploded because like the leaf doesn't look anything like I thought it did, right? I thought a leaf looked a certain way. I thought it looked like this, right? And it doesn't look like that. And I'll show you in just a minute what it actually does look like, right? And so we can pull things out like the total surface area of the leaf. We can actually measure it now in a way that's never been before possible. And we can measure the total amount of airspace, and we can talk about things related to the distribution of that airspace and why it's important. And we can pull out things like the vasculature as well. So pull out these different components and think about them critically in trying to figure out why a leaf looks like a leaf. All right, so I'm going to do this, and I hope it works. All right, so what it looks like, and I will do this. We're going to move over. Okay, it worked. Awesome. All right, so this, anybody eating lunch? Eating a sandwich? Or ate a sandwich? Who had spinach today? Ate a piece of spinach? Nobody? 1, 2, 3, 4, a few people. How many people have eaten spinach? All right, everybody. All right, so this is a common species. All right, so this is what the inside of a spinach leaf looks like. All right, so you've seen spinach from the outside, certainly. But this is what a piece of spinach looks on the outside. So this is top to bottom, so I've flipped it over intentionally. So this is the top of the leaf, and this is the bottom of the leaf, and I'll tell you why I did that in just a minute. These are the palisade cells, which are those long column-shaped cells, and these are the spongy mesophyll cells that we were just talking about. And certainly, if you were to look at just this face that sort of you're looking at, and these kind of darker outlines, I can totally understand how you would arrive at the sort of generic little spheroid sort of spongy mesophyll paradigm that we see. But the first thing, and I hope this will work, is that we can zoom in and we can look at this leaf from the inside, and we can fly in, right? And so you can see that the inside of the leaf is much more complicated than we originally thought. And so we have no idea at the moment what this diversity actually means, what it actually looks like, and how much diversity there's in the inside of the leaf. So if you look at all those leaves that I put up on the screen, all those different species from E.L. Myers, there is that diversity on the outside, but there's also this incredible amount of diversity on the inside that is equally important. So in terms of conservation, perhaps, we want to conserve a particular species because it plays a particular role in the habitat. But there's all this amazing diversity on the inside, and the degree of plasticity we are still trying to wrap our heads around and trying to figure out what it all means, right? So this is all very new. We've now at least doubled or tripled the number of people that have now seen what the inside of a leaf actually looks like, right? Just by sitting in this room, because this is so new. And my students actually in my plant physiology class just got to see this yesterday, and they wouldn't put the thing down. They wanted that. Some of them ended up being late to class, I think. But it ends up, to their next class, right? So you look around and you spin this thing around, and one of the things that we'll do, I'll show you how you can do this and play around with this at the end of this seminar, right? So that's what the inside of the leaf looks like. All right, there we go. We're back. Cool. All right, so back, enough spinny things. So now we can measure things like leaf porosity that were otherwise really, really difficult. There's ways to do it before, but it was really, really hard, and there was potentially a lot of measurement error that we weren't quite sure of. So a lot of uncertainty, and so I think we're now a little bit more certain about a lot of these measurements, right? So we've got cross-section of the leaf. This is a micro-CT scan, just a single slice. We've got cells, we've got airspace, we've got the stomata, and we've got all the different parts, and we can look at it in 3D, these three-dimensional volumes, and we can figure out where all this stuff is. We can look at leaf tissue, we can look at the airspace and kind of how the airspace is distributed, and we can actually measure the surface area of the leaf for a given volume instead of a cross-sectional area, which is a really important major advancement in our ability to understand plants. And so now instead of looking at it in square microns per square micron of tissue, we're looking at it, as how much surface area is on the inside for a given volume of tissue. We can also measure all sorts of other things, right? So we can measure the percent of the airspace that's disconnected from the main airspace, and we'll talk about this in a minute, why this is important. We can also talk about path length and tortuosity of this pathway, of the vapor diffusion pathway, either CO2 getting to the cells, or the photosynthesis is taking place, or the water vapor that's coming out, and do this sort of in real time and do some really interesting modeling exercises that I'll get to at the end. But this is kind of a major advancement for us as well, because these terms had always been sort of assumed or not, it's just things that you could not measure before we started to do this. All right, so what we wanted to do is think about just a simple hypothesis and look at two different groups of plants, CAM plants and C3 plants. So C3 plants are the ones that do photosynthesis like most of the organisms that if you walk outside, most of the plants out there are doing what's called C3 photosynthesis. It's a very common biochemical pathway that plants use to do photosynthesis, and it's dependent on having a really high absorption rate of CO2, and as a consequence, a really high water loss. CAM plants do things a little bit differently in that they will accumulate CO2 at night, and then store it away as a different molecule and then use it later on during the day. And these plants are typically, and we're going to use bromeliads, the bromeliad group, as a way to make these comparisons, because we have C3 plants and CAM plants both in the same group and very closely related, and then make some comparisons between the two. And so we would expect that the C3 plant should have a high surface area, lots of airspace to optimize CO2 uptake, and CAM plants should have a lower surface area, less airspace in order to optimize CO2 storage. They need to have really, really big cells to store the sugars, or the sugar analog molecule, in order to store it for a long period of time, and then also to minimize water loss. They want to do the exact opposite of C3 plants. They're going in a really hot environment without much water. They want to be as careful with their water as possible, and so they want to do the opposite of what a C3 plant does. And basically, what we wanted to do is go in and figure out what does this vapor diffusion pathway actually look like, and calculate all the surface area. Right? So you can take a leaf. This is achmea. This is just a piece of it, right? And you can kind of look at it and spin it around. And what this one is going to do is you can see how complicated. Again, this doesn't look anything like our kind of generalized cartoon from the biology textbooks or even the botany textbooks. We're going to go through, and we can fly through one of the stomatopores, and we can think about what is the kind of the maximum pathway, what is the path length, and how tortuous is that pathway for getting to where the CO2 might be absorbed on the inside of the leaf, because that is really important. That is going to be the fundamental trait that determines whether or not, if the plant can make this pathway as efficient as possible, then it's going to be able to pull in more CO2 and do better at photosynthesis. All right. So we took 19 different species, 19 bromeliads, all in the same plant family. Lumped them into two different groups, CAM plants and C3 plants, and then basically had to use our software and teach it plant anatomy so that it could go through and automate a lot of this process in picking out the intercellular airspace in white, the vascular tissue in the bundle sheath, which is just basically the veins, and then all the rest of the cells, the mesophyll cells. And this is just a single slice to illustrate kind of what's going on. And one of the things that's immediately apparent is that these C3 plants, right, have this huge, really large, open central airspace, and then the CAM plants have the vascular system sort of distributed in all sorts of different ways, and we think that at least this is in part because bromeliads often don't follow this flat green sheet, right, paradigm, right? So they've deviated that away from that in order to adapt to these really dry, high light environments, right? And so what we can do is then we can take the leaf, right, and all these little arrows point to the stomatal inlets on the bottom underside of the leaf, and we can start to calculate things like the surface area on the inside, or we can talk about the vapor path length. So basically, how long would it take for a CO2 molecule entering the leaf to get to where it needs to go, right? So it's going to have a longer and longer path length, and the length of the vapor path length is going to directly influence the plant's ability to uptake CO2. And what we find in the CAM plants in particular is because the cells have gotten so big, right? So they get bigger and bigger and bigger, basically squeezing out all the air, right, and all the airspace, and that's disconnecting a lot of these little airspaces in the top of the leaf and making it really hard for any of the cells near the top of the leaf to do any photosynthesis. And indeed, when you look at as the vapor path length gets longer and longer and longer, the leaf actually stops putting chlorophyll, stops putting chloroplasts in these cells, probably because the amount of CO2 that it can actually reach because of that 10,000 times slower diffusion gradient penalty they basically stop. They use these for water storage, and then they stop putting chloroplasts in them because there's just not enough CO2 to make it worth it. Because all those cells on the inside, each of those cells on the inside of a leaf, to keep them alive, there's respiration and there's a cost involved in doing that. So we can take our 3D data and then collapse it back into 2D and produce these kind of heat maps and basically look at all of these different traits of these different CAM and C3 plants and start to look at them in very new ways. And if we look at the porosity of CAM versus C3 plants, the porosity is way higher, right? So if you look at all of these CAM or C3 plants down here, the need to optimize that surface area to volume ratio, they seem to be doing it, right? A big internal airspace, and there's very few parts of the leaf that are actually disconnected from the main airspace. And then CAM plants are doing something very different, right? So there tends to be like a little pocket, and then there's a lot of the leaf is actually disconnected. And this is this disconnected airspace, the closer to the white color that you see on there, the more disconnected the airspace is, and then functionally renders those parts of the leaf non-photosynthetic or very, very inefficient. And so in the case of CAM plants, that's probably okay. They have relatively low photosynthetic rates. They're just trying to get by in a really hot environment, right? And so two very different strategies for living in a dry habitat, and because they're living essentially in the same habitat, but they've gone through evolutionary time, have diverged into either being a CAM plant with a very different biochemical pathway, and so have the form of the leaf then looks very different in order to maximize or optimize the photosynthesis, doing CAM photosynthesis, doing C3. And we look at some of these other terms, the two different groups, the C3 plants and the CAM plants, sort of separate out into these two different groups. If we talk about tortuosity, we talk about lateral path lengthening and airspace connectivity, the two different groups fall out into two different kind of trait spaces for all these different parameters in terms of the mesophyll surface area, which is really interesting and sort of what we expected in this case study. And what the really interesting thing about CAM plants is that we think that they're probably they've done the absolute best and sort of what's the sort of evolutionary trajectory for these particular traits. And it looks like, so C3 plants are kind of in this domain in terms of porosity. This would be a porosity of 0 where all the airspace is squeezed out. You never see plants that look like this because that just would be a very bad way to put together a plant. And this is where the pore space is all entirely connected, but these leaves are sort of floating in three-dimensional space and not connected to it at all, and this probably wouldn't arise unless you were maybe growing plants in space or something like that, but even then it still wouldn't work. Most plants fall in sort of this range between 0.12 and 0.5. So this is sort of the realm of reality from here to here, and this is where the C3 plants are located, and then the CAM plants are over here, where they've basically squeezed out as much of the airspace as absolutely possible. And this is a simulation of the total amount of malic acid, or that compound that CAM plants use to store away the carbon until they can actually the CO2 until they can actually use it. And the maximum rate happens to fall based on our model predictions right in that zone. And so C3 plants are probably, in terms of the clustering of the cells and how large the cells are, and putting together the inside of the leaf in terms of the surface area to volume ratio, perfectly optimized for doing CAM photosynthesis. And then the C3 plants don't exist sort of in this porosity domain simply because it would make them a terrible C3 plant, right? Because they need to do something completely different with maintaining their surface area to volume ratio. So that was really cool. And then what we started to do is we went back to this very disappointing data set that I mentioned earlier, looking at surface area in terms of meters squared of micrometers squared of surface area per micrometer squared of of leaf that you're actually looking at, and then looking at the maximum photosynthetic rate. And then we move it over here, and we actually measure it on a per volume basis, measuring the trait in 3 dimensions. And then all of a sudden, we get this really great relationship. So this is an R squared of 0.25. If you're in biology and ecology, and you're able to get an R squared of 0.25, so basically we can now explain 25% of the variability in the maximum photosynthetic rate with one trait, right? So mesophyll surface area. Right, so, which is really amazing. There's a paper published in Science like last year. They had an R squared of 0.28 or something like that, right? Because it was just talking about leaf size and sort of patterns, global patterns in leaf size. So you get really, really excited about an R square of 0.25, and there's more data to add to this, and it's going to get better and better, right? So we can kind of now explain and sort of fill in that missing part. Right, so if we look at the area of the mesophyll for a given volume of mesophyll tissue, we look at the angiosperms, and we can look at the gymnosperms, and we can look at the ferns, and sort of breaking these into 3 different bins. The angiosperms have explored this huge amount of trait space, right? So they go from really, really low surface area to volume ratio to really, really high. And the gymnosperms, right, so these plants that were historically dominant on the planet are evolutionary kind of in a dead end. They're stuck. For whatever reason, they haven't been able to break out and take advantage and optimize the surface area to volume ratio, right? And there's even some of the ferns have started to play around with this, and there's these xeric ferns that live in dry habitats. They look a little bit more like an angiosperm. And then one of the interesting things is if you break down the angiosperms and look at this trait diversity, you look at cam plants, and they're low, right? That's what you'd expect based on the sort of model simulations that we just showed, and that cam plants want to have a really low value here. And then the C3 and C4 plants are the ones that are making up a lot of this diversity. And then if you look at just the monocots and break those down into cam versus C4 and C3, you see this huge range in diversity. And in particular, these C3 and C4 grasses have just kind of exploded in trait space. And we think that's one of the big reasons why they've been able to become dominant in almost every terrestrial habitat on Earth, right? And out-competing all these other species, right? So we've got this. We think that surface area, mesophyll surface area, like the breadth or the diversity in mesophyll surface area has also gone up over this evolutionary time. But then Mason and Guillaume and I were thinking about it, and then how did they do it, right? And so if I ever have a question now in the lab, after working with Adam, Roddy is a Yibs postdoc in the lab. I ask Adam. All right, so ask Adam Roddy is the thing to do, because Adam has a really cool way of thinking about the world, and one of the great things about working at FES is that you get to work, interact with all these really amazing people. So I asked Adam, what do you think? And he said, oh, it's genome size or cell size. And Adam had this really cool paper with a colleague at San Francisco State University last year that's sort of what I think is underappreciated. And what they found is this really cool relationship between the size of the genome, the size of the DNA, right, that's in each and every one of the cells, and the maximum and minimum cell size. And so by having a smaller and smaller and smaller genome, by streamlining the genome, that means you can have a physically smaller cell size. So cell size, so smaller cells, basically means greater surface area for CO2 uptake. And indeed, if you look at the surface area to volume ratio and look at mesophyll cell diameter, right, so there's a really strong relationship between these two. So as your mesophyll cell diameter gets smaller and smaller and smaller, your surface area to volume ratio skyrockets, right? It allows you to absorb so much more CO2 from the atmosphere and do much more photosynthesis. And if you look at this in terms of genome size, all of the plants that have really small mesophyll cell diameters are species with small genomes, right? And so this sort of idea and this clustering of ideas pairing up in just this big question mark sitting in the middle. And we think it's controlled by cell size. The smaller the cell size, the more surface area to volume ratio you can have, and optimizing that. And ultimately, that is probably determined by genome size. And genome size is something that we can probably manipulate in the future, right? So if we're thinking about gene editing and pulling out all the parts of the gene that don't matter, then maybe we can get cells to produce to get leaves to produce cells of smaller size, or then we can start to play around with things and then maybe cause plants to have higher productivity, higher yields in our crop species, and things like that. But wait, there's more, right? So it doesn't stop there, right? So that was cool, but there's more. And I just got done telling you about how most plant physiologists are sort of siloed into their own world, right? And we just talked about the fact that the leaf is being pulled in all sorts of different directions from a functional perspective and kind of morphological perspective. But what's also interesting is that we don't know, so there's got to be some trade-offs, right? So if a leaf goes through all this effort to basically increase the surface area to volume, right? So what else is surface area good for? And this is a project that I wanted to highlight really briefly. It's sort of in its initial stages. This is work that's been done by Alika Borsuk. She's an MESC student in my lab right now, and is working on the optics of these different tissue types. So I remember we've got those palisade cells that look like long, skinny cells, and we've got these in the palisade tissue in the upper parts of the leaf, and in the lower parts of the leaf, where all the surface area that we've just been talking about in these small cell sizes are located for the optimizing CO2 uptake. Well, what does that mean for processing light, right? Because the leaf has to also process light, and it's going to be influenced by the shape of the cells. And so Alec has been working on some really cool stuff. We're simulating basically the rays coming in and doing this for the first time in 3 dimensions and figuring out, well, there's been some theories about how these different cell types perform in terms of transporting light through the leaf. And what she's found is that indeed this theory holds up, where the palisade cells tend to act as kind of light pipes and channeling light deeper and deeper into the leaf, and then the spongy mesophyll cells sort of act as light diffusers. So not only does decreasing the cell size increase the ability of the plant to pull CO2 out of the atmosphere, but it also influences the ability of the leaf to sort of scatter light and trap as much of it as absolutely possible. Right? So thinking about things in terms of not just siloing yourself in one kind of field, and sort of thinking about leaf form and function from a bunch of different approaches based on kind of these competing demands for why a leaf might look like a leaf. All right? So now what she's doing is taking these 3-dimensional CT scans. These are sort of models, modeled kind of toy leaf cells, and then actually pulling out the actual cell structure on the inside of the leaf, and then running these simulations to figure out whether or not, and testing some of these hypotheses. Okay, so if you have a leaf that has a really thick palisade layer with these cells, what does that actually mean? Right, so we're kind of pushing into these new areas that basically have never been before possible because we just hadn't had access to kind of the complexity of the internal geometry of leaves. And then in terms of biomechanics, so what does it mean, right? So a leaf has to be flexible, it has to be rigid, it has to hold itself up. So if you start to shrink cells and move them in different directions or have really big cells, what does that do for biomechanics, right? So if you look at other examples in nature, right? So this is an old image from a metacarpal bone from a vulture's wing, stiffened after the manner of a Warren's truss. Right? And so this is the inside of a vulture wing, and in order to, we know that birds have really, really lightweight bones, and that of course allows them to fly, right? And so basically taking advantage of some of these mechanical terms and working with a group from the School of Engineering, and Adam as well, Adam Roddy again, working through this Gibbs-funded EF3 program, looking at the evolution of flower form and function, and also in relation to leaves. We're starting to look at things, and so what are the biomechanical implications of putting together a leaf in this way? Right? So there's all sorts of different things and compromises and constraints. So we think that the increased mesophyll surface area, so probably increases the water vapor movement, right? So water out, and that means that it also increases the CO2 absorption, pulling CO2 in. It optimizes the internal light scattering, perhaps. We're testing that right now, and there's probably a role in leaf biomechanics. And in this way, we think that we can now produce a new sort of integrated view of the 3-dimensional leaf, both from the intracellular organization, because now we can do this micro-CT imaging or 3D imaging at the inside of the cell, like inside of individual cells. We can do it for all the things that we were just talking about into the intracellular level, and then the tissue level organization, and kind of thinking about things in a very integrated way. We're also doing this in virtual reality. This is, and looking at things just like you did with looking on the screen. We're exploring leaves in virtual reality. This is Graham Farquhar. Those of you that know anything about plant physiology know this guy's name. So he's a Kyoto Prize laureate, and one of the people responsible for kind of the primary equation that we use for estimating photosynthesis. So he's in here. If you don't get excited about Graham Farquhar, you might get excited about Secretary Rick Perry, because the facility that we use is funded by the Department of Energy. And so Rick Perry came out and did a site visit. That's our poster in the background of some of the work that we do. And there's Rick Perry with the 3D virtual reality goggles, and he's looking at one of our samples, right? So he's in there. And I think one of the things that really came out from Justin's talk last week is that we're living in this time of increasing sort of information and misinformation. And it's our job as scientists to do a really good job of communicating what we find is important about plants and sort of diversity, or in terms of biofuels that the Department of Energy is interested in, and then funding this large-scale facility. The fact that we can get Rick Perry to come and sit down and put on a 3D pair of glasses and look at the inside of leaves or look at the inside of plants, I think is really powerful, right? Because these are the people that end up making the decisions that influence the rest of us, right? Okay. And so what I want to do, and the last thing that I want to leave you with, is that if you liked that 3-dimensional leaf, one thing that you can do is if you have an iPhone, you can go here and you can open your camera app, at least in the newer versions of the operating system, you can scan this. It will automatically detect this QR code, and it'll load up a website that has this, and you can actually, everybody's got their phone up. I should take a picture. So you can actually download, go to a website that will allow you to download this model, and you can pinch and zoom and zoom in 3D and look at this on your own time. And I can send out a link, or we can figure out a way to get it. And if you're ever interested, feel free to walk by my office and stop in and say, hey, I want to look at the inside of a spinach leaf, because I've got a virtual reality headset just sitting around waiting for people to play with it. We were using it in class yesterday. It was a blast, and we're doing this all the time in my classes. All right, so thinking back to the lungs, right? And that's like a very, very personal thing. It's something that for all of us. And I did a back of the envelope calculation this morning, and I think I'm right. And so this red maple leaf that we started off at the beginning of the lecture is about 40, maybe 40 square centimeters in surface area. And that would have, if based on our new calculations, would probably have about 1,400 square centimeters of internal surface area, right? And so that one leaf, right, might have 0.14 square meters of surface area on the inside. If you take, which doesn't sound like a lot in terms of a pair of lungs. But if you multiply that for maybe a medium-sized tree of about 1,000 leaves, you get 140 square meters of total surface area on that one tree, which is an amazing amount, right? And it's about the size of a tennis court, which is pretty cool. All right, so I've got a lot of acknowledgments, a bunch of people to thank. There's not enough time to do it. And I just wanted to thank my lab in particular. All the great collaborators at UC Davis and at Berkeley are funding from YIBS and the National Science Foundation and the Department of Energy, and a lot of other great people. Kevin Boyce at Stanford, who helped out with kind of a lot of the paleo work and kind of thinking, and Dan Johnson, of course, who's also part of one of my other NSF grants that I didn't have a chance to talk about today. But I'll leave you with that, so thank you. So I'd be happy to take any questions if I know we're just about out of time. Shuei.
So earlier you implied that diffusion efficiency difference between air and water may have played a role in shaping the morphology of a leaf. And later on, you bring in can plants and showing the contrast between can plants and C3 plants. There's a group of plants growing in water, aquatic plants growing underwater that also are can. And of course, they have very low diffusivity is a huge obstacle, right. But they don't need to conserve water. I wonder if you can explain, their leaf morphology are almost like the same as we see on dry plant leaves.
Yeah, so there's one of the things, and what's hard to tease apart from all of this is kind of understanding sort of the evolutionary history about how those plants evolved. Did they start on land? Did they migrate back into the water? Or did they stay in water the entire time? And so there's, in a lot of these species, there's a lot of evolutionary kind of baggage that goes along with it, and trying to tease that apart can be generally pretty hard. And certainly growing underwater is difficult in terms that you just mentioned, in terms of bringing in CO2. One of the things, so we did measure kind of the maximum photosynthetic rate and plotted that against the surface area to volume ratio there. And what I want to point out and what I want to make sure that is clear is that maximum photosynthetic rate isn't the absolute trait that you need to win and to outcompete, right? So in many cases, you just need a photosynthetic rate that's good enough, right, to outcompete all the rest of your neighbors. And so there's probably some trade-offs between there. There's other structural things that an aquatic plant would need to do, and there's therefore some sort of a compromise in the way that a leaf is put together. So I don't have a good answer for your question, but I tried to dance around it there and sort of. Anything else? In the back.
So to me, one of the things that jumped out the most in comparing like the 2D versus the 3D image of the leaf is that in a 2D cross section, the spongy mesophyll looks like round blobs, but in this, it looks more like it's kind of stringy. So I was wondering, is that changing our picture of the shape of spongy mesophyll cells, or are those sort of like strings, actually several cells stuck together? But either way, like, what do you think is the significance of that structure?
Yeah, so there certainly are some plants that have the kind of the spheroid paradigm, right? So a lot of those can plants, it turns out, they look pretty sphery, especially in the top part of the leaf where the water storage is going on, where they really need to just be a big bag of water, essentially. But the rest of these, but even in those species, even in those succulent plants, when you get down into the spongy mesophyll, it starts to look really weird. I don't have a good answer for you because we've just started looking into this, and this was the first one that I really started to poke around in and spend some time kind of thinking inside the leaf, and it's just not what we thought. So I don't know. I don't have a good answer for that. We're sort of working on it. The inside, Adam has been doing a lot of work on flowers, and it turns out the inside of flowers looks just like the spongy mesophyll. There's no palisade, and they're kind of doing very different things. And so we're thinking about that in terms of kind of the biomechanics. Certainly there's a couple of spots where you can see there's a cell wall there, there's a cell wall there. And so the mesophyll cells don't, at least in a lot of the species that we looked at, don't look anything like the textbooks. But we wouldn't have known, right? So the only way that you would know is by doing thousands and thousands of cross sections by hand and sort of reconstructing it in your mind. And it's a very painstaking, tedious task to do that, right? For one leaf and sectioning it perfectly thousands and thousands of times. It just is really something's really difficult to do without this technology.
So another person that believes in biomimicry. Who wants to get inspiration from this leaf. And I want to fix CO2 and transform CO2 and do it selectively aside from all the other gases and things like that. What is this leaf, what are these insights teaching me on how to do it better?
Right, so in this case in particular, just because given that ratio, right, that exchange rate of 400 to 1, there may be some things that, if we were able to manipulate this in a certain way, just by tweaking that surface area to volume ratio, we might make a leaf that ends up leaking way too much water and becomes a bad plant. It doesn't do it the right way. And so certainly from this perspective, or from the perspective of the lungs, that's just one of this kind of underlying biological principles is the surface area to volume ratio. So if you can maximize that, so if you're thinking about trying to pull CO2 out of the atmosphere, which plants are really, really good at, then this is, this type of kind of organization, and in particular the species that are really on that far end of that trait space that have really maximized this is sort of where you would want to go, right? But it may not be this. It may be some sort of porous media that you might want to go at. But at least in evolutionary terms, plants have been thinking about this. There's 400 million years of research and development, and so that have gone into putting it, turning a leaf into this. And so we should probably start paying attention. Yeah. Mm-hmm. Okay, I think that we gotta wrap it up. I think somebody told me we had to stop. All right, thank you. Hey, thanks. Yeah.
Plants have this enormous phenotypic plasticity.
They do.
They can change the size of the stomachs.
Right.
In the right conditions, they make large stomachs. Other conditions, small stomachs.
Yeah.
So I don't know what that all means, but
I don't know. Well, I mean, so like given, I'll turn this off.